This is the second video on inheritance, and in this video we're going to talk about individual variation that occurs from genetic recombination or mutations or chromosomal abnormalities. You are not a 50-50 mixture of your parents. During meiosis, the maternal and paternal chromosomes are randomly distributed, so each gamete has a unique combination of maternal and paternal chromosomes. Only in rare cases will an individual receive both alleles from one parent. This might occur in amphimixis after the secondary oocyte has been fertilized. If the maternal chromosomes don't separate during meiosis II, as the secondary oocyte becomes an ovum. During meiosis, various changes can occur, crossing over or genetic recombination. These cause great variation among gametes. During that synapsis, tetrads form and adjacent chromatids may overlap. The chromatids may then break and some of their segments trade places. This is called crossing over. During genetic recombination, some of these segments trade places with a non-homologous chromosome. This is called translocation. During genetic recombination, portions of chromosomes may also break away and be lost or deleted. This is abnormal and can have severe or lethal effects on the zygote. Recombination that results in abnormal chromosome shapes or numbers is lethal for the zygote. This occurs in 10% of all zygotes. Chromosomes are damaged, broken, missing, or there are extra copies of the chromosomes. If large numbers of genes have been added or deleted, the mortality rate and the severity of the problems increase. This may happen in women who become pregnant later in life. The longer the oocyte remains suspended in meiosis I, the more likely the recombination errors will occur when meiosis is completed. Amniocentesis is a procedure where a sample of amniotic fluid is removed and the fetal cells it contains are analyzed. Many genetic conditions can be detected this way. A needle is inserted into the amniotic cavity and a fluid sample is obtained using an ultrasound as a guide. This poses a potential threat to the mother and the fetus. Amniocentesis must be performed when there's enough amniotic fluid to avoid hitting the fetus. This usually is between the 15th and 18th week of pregnancy. Embryos that have abnormal autosomal chromosomes rarely survive. Translocation defects and trisomy are two types of autosomal abnormalities where the fetus does survive. Translocation defect is where an exchange occurs between non-homologous chromosomes. For example, a piece of chromosome 8 exchanges for a piece of chromosome 14. The genes that move may function abnormally, becoming inactive or even overactive. If there's no net loss or gain of chromosomal material, the embryo may survive. For example, if chromosome 8 moves to chromosome 14, but chromosome 14 doesn't leave, then there's a higher chance of survival. In trisomy, a mistake occurs in meiosis. One of the gametes involved carries an extra copy of one chromosome, so that the zygote then has 3 copies of this chromosome instead of 2. Individuals with trisomy 13 have 3 copies of chromosome 13. Zygotes with extra copies rarely survive. One exception to this is trisomy 21. Trisomy 21 is also known as Down syndrome. It's the most common chromosomal abnormality. 1 in 700 babies are born with Down syndrome, with different ranges of developmental disabilities. Some individuals with trisomy 21 live to a moderate old age. Many, however, develop Alzheimer's disease while still young, before the age of 40. There is a direct correlation between the mother's age and the risk of having a child with trisomy 21, meaning the older a mother is, the greater the risk of having a child with Down syndrome. Mutations are changes in the nucleotide sequence of an allele. A spontaneous mutation results from random errors in DNA replication. These are common, but in most cases, the error is detected and repaired by enzymes. If undetected or unrepaired, they can change the phenotype in some way. Mutations during meiosis can produce gametes with abnormal alleles. The alleles may be dominant or recessive. They may occur on autosomal or sex chromosomes. Most of the mutations make the zygote incapable of completing normal development. Mutations are the primary cause of high mortality rates among pre-embryos and embryos, causing miscarriages. Approximately 50% of zygotes fail to complete cleavage, and another 10% fail to reach the 5th month of gestation. If the abnormal allele is dominant and does not affect survival, the phenotype will show the effects of the mutation. If the abnormal allele is recessive and on an autosomal chromosome, it will not affect the phenotype as long as the allele from the other parent is normal. An individual who carries a recessive autosomal allele and doesn't show the effects is called a carrier. If that individual and another individual with the recessive autosomal allele produce a zygote homozygous for the abnormal allele, they will show the phenotype. An example is Sachs disease. If both parents are carriers of the autosomal recessive disorder Sachs, they have a 25% probability of producing a child with Sachs disease. Sachs affects chromosome 15 and destroys nerve cells of the brain and the spinal cord. The presence of a particular pair of alleles does not affect the phenotype in the same way in every individual. Penetrance is the percentage of individuals with a particular genotype that show the expected phenotype. For example, emphysema has been linked to a specific abnormal genotype. However, 20% of individuals do not develop emphysema even if they have the gene. So the penetrance of emphysema is around 80%. Expressivity is the extent to which a particular allele is expressed when it is present. If a given genotype does not affect the phenotype, it can do so in varying degrees depending on the activity of the other genes or environmental factors. For example, identical twins have the same genotype, but they do not have exactly the same fingerprints. Environmental effects can be very evident during embryonic and fetal development. Drugs such as antibiotics, alcohol, and nicotine from smoking can disrupt fetal development. These factors that cause abnormal development are called teratogens. The entire karyotype is called the human genome. It is the full set of genetic material or DNA in our chromosomes. From this human genome, 20,000 to 25,000 protein-coding genes are present. From these genes, there is a recorded 10,000 different single gene disorders. Most of these disorders are very rare, but collectively they affect 1 in every 200 births. Genetic screening for abnormal genes is now performed for many of these disorders. This ends our second video on inheritance for Chapter 29.